So we don't classify this as simple columnar or simple cuboidal, it's glandular epithelia, all right? So, on this view, it should be fitting like this, but of course the stand covers up the anterior aspect of the lobe. So I'm going to tilt it down. We have the large lobe, which is the right lobe, and then next to it, the left. And then posteriorly, we have two. These are shown at right angles, although in most diagrams, they're shown as if they were on the same plane. So between the left lobe and the right lobe, to the left of the gallbladder, we have the quadrate lobe, 4 sides, okay? So it's bounded by the ligamentum teres on one side and the gallbladder on the other, and this would be the quadrate lobe. And then on this one, we have, next to the inferior vena cava, another lobe that has 4 sides. We can't call it quadrate again, but it's on the posterior aspect. All right, so here's the inferior vena cava, and this one is bounded by the ligamentum venosum and the inferior vena cava. And so our word for posterior sometimes is tail or caudal. All right, so this is caudate lobe. So on the inferior aspect between the gallbladder and the ligamentum teres, we have the quadrate. And then most posterior between the ductus, the ligamentum venosum, and the inferior vena cava, the caudate. Okay. So something that we didn't really mention with the ligamentum teres is another and our last mesenteric structure. I can see a good, don't think I have a good visual of it, but right here, let's see if I can kind of get a view of it. So here would be the anterior abdominal wall, and we have the umbilical vein, which is carrying oxygenated blood, coming in from the placenta and traveling to the liver. It's a little larger than actual, just so you can see it here. The gallbladder is usually drawn green, but I'll draw the whole thing green here. All right, so this is the, we'll call it the postnatal structure here. The ligamentum teres, which is our collapsed umbilical vein. It travels in a mesentery. So we have this mesenteric structure. We have the parietal peritoneum of the abdominal cavity. And then we have this mesenteric structure that essentially suspends the ligamentum teres. And that's what we see right here. Okay? So this cord-like structure right here is the ligamentum teres, the collapsed umbilical vein. And then that mesenteric structure, if I were to draw a transverse view, here would be the collapsed umbilical vein, okay? And then our mesenteric structure would be the parietal peritoneum. And this is moving superiorly towards the liver. So this structure right here is known as the falciform ligament. That's the mesentery. So it's a double layer of peritoneum that supports the umbilical vein from the belly button to the liver, where it runs into the liver to supply oxygenated and nutrient-rich blood to the liver until it collapses. Alrighty. So now let's look, any questions on this? Unfortunately, most of our models just show the ligamentum teres, okay? But imagine it's suspended about an inch or so from the deep surface of the anterior abdominal wall. All right. So the liver is made up of epithelial cells known as hepatocytes. The liver is the largest gland in the body. As you realize from our blood discussion, it gets first dibs at the nutrients that are absorbed from the intestines. Hence we have a venous portal system. The capillaries within the villi of the small intestine are capillary bed 1. Then they form superior mesenteric vein, which goes into the hepatic portal vein and join with the hepatic artery, and we have our sinusoids, which are capillary bed 2. So the liver is formed of tiny microscopic 6-sided structures known as lobules. In the human, we do not have this amount of connective tissue, so it's hard to see the 6 sides. This is from a pig, so there's a thick amount of connective tissue around each 6-sided lobule. It's kind of hard to draw dimensionally. Bless you. Yeah, erase the board. So as both the hepatic artery and the hepatic portal vein come in together and form the corners, one, two, three, four, I'm getting 5 sides here. We'll put another one here. Maybe it's a little off sides. So blood is coming in and branching out to form branches, although we tend to leave that word off, of the hepatic portal vein. Okay? This is one of our hepatic lobules. This structure would be shared with another hepatic lobule, as you can see here. Okay? So each of these corners is going to be shared with between 3 different lobules. Then our other vessel to come in, so the portal vein is rich in, as we'll see when we talk about absorption, this is going to be rich in amino acids and monosaccharides. We're missing lipids, triglycerides and fatty acids, because those to travel in the blood have to have a protein coat to make them soluble in the blood, because otherwise they're nonpolar covalent. And the large protein coat that they pick up crossing through the simple columnar cells of the intestine make them too large to move into the blood vessels. So they move into the lacteal. Remember, those are large enough for white blood cells? So they're large enough for these protein-coated lipids. So the hepatic artery is bringing in lipids and oxygen. And it flows side by side with the portal vein. Now, those 2 fluids, the venous blood coming through the branch of the hepatic portal vein and the arterial blood coming through the branch of the hepatic artery, mix as they flow by the liver cells towards the center of our lobule. So we have a mixture. These are sinusoids, which are very large capillaries. Remember, capillaries are typically about the diameter of a red blood cell. So that the red blood cells have to flow slowly, single file, allows time for oxygen to move off of the hemoglobin. Well, here the purpose is more for some oxygen distribution, but also for the nutrients. And so as these vessels, I need a purple one, flow together, they form these channels known as a sinusoid. Okay? And this is occurring up and down the entire level. So there are horizontal branches that connect the portal vein branches and the hepatic arteries. And then from those all up and down, think of a stack of bicycle wheels with wire spokes. All right, instead of being nice and round, it has 6 sides, so it's not going to roll very well. And those spokes would be sinusoids. All right, so the entire up and down height of the hepatic lobule, we have these sinusoids flowing towards the center, where they all, once they've given up their some of their nutrients, not all, because they're going to go to the other organs, and this central structure here is known as a central vein. All right. So if we look at the sequence of flow, and I'm going to write this with blue just because my black is not very dark. So picking up the nutrients from the small intestine, we have the capillaries. And lacteals within the small intestine, okay? Now I'm not going to follow the lacteals all the way back to the subclavian by writing it down, but if you recall, they would be going to lymphatic vessels and trunks, to the cisterna chyli, to the thoracic duct, to the left subclavian vein, the brachiocephalic vein, the superior vena cava, back to the heart, out the abdominal aorta, and hepatic artery, okay? So now, capillaries are going to enter the superior mesenteric vein. And then that will flow into the hepatic portal vein and its branches. The lacteals will eventually get into the aorta and the hepatic artery, and that will come together with our branch of the hepatic portal vein. Okay. At this point, the two components flow together as a sinusoid. All right. So this is cap bed 1, and the sinusoid is cap bed 2. This is not an arterial portal system, it's another venous portal system like we had in the brain. So the sinusoids then flow together to form the central vein. And you know what comes next, because we've talked about it already. What is the vessel that carries blood out of the liver? Hepatic portal vein carries it into the liver. Hepatic vein, all right? So central veins then flow into the hepatic veins. There's usually between 3 and 4. And hepatic veins then flow into the inferior vena cava. Okay. Now, notice how thin. You can think of these as plates of hepatocytes forming a radiation around the central vein. Notice that those plates of hepatocytes are never more, and I realize this is an artistic drawing, are never more than 2 cells thick. So each hepatocyte is going to face a sinusoid. All right? It's going to have access to the content, the amino acids and the sugars and the lipids flowing through that sinusoid. So essentially, I'll erase one of these corners here. Well, I'll do it here. So looking down on the top or the side, which way you will. So this is the hepatocyte. You would have a sinusoid would be exposed to. And notice I put a gap in the middle. There's no epithelial separate lining. What lines that channel is just the plasma membrane of the hepatocyte, all right? So essentially what is occurring there, let me fill in the nucleus so it doesn't look like it's another gap. So essentially what's occurring there is a channel created where the bottom part of two cells separate. And we line a whole bunch of cells up, and we have a tiny little channel or canal or canaliculus, all right? So these green structures, channels that you see here, are the bile canaliculi. And they're carrying bile and bile salts in the opposite direction. So the hepatocyte is going to dump, and because we generally see bile as green, hepatocytes are going to, from their basement membrane, and we'll look at the role of bile salts on Wednesday. And the bile canaliculi flow in the opposite direction to empty into bile ducts. You'll notice I drew three structures up there. A branch of the portal vein, a branch of the hepatic artery, and a bile duct. Okay, the bile canaliculi running in to form the larger bile duct. The vessels are lined with what type of epithelia? Simple squamous. Okay, the bile duct is simple cuboidal, which is going to have a larger muscular wall. The hepatic vein branch or the portal vein branch or the hepatic artery branch, which is going to have a thicker, smooth muscle wall for its diameter. The artery. That's how we tell the difference on a histology slide on what is what. This is just showing a diagram of the lobule. All right, so here's the corners are the branches of the hepatic portal vein and hepatic artery. Here are the sinusoids going through, and this would be, actually this is in the center, that would be the central vein. And we have a portal triad. Branch of the hepatic artery, branch of the portal vein, and a bile duct. Okay?
You said bile duct is simple
Cuboidal. I'll come back to that in a moment. So here are some histology slides. This is showing the sinusoids. So we're seeing hepatocytes. That is lined by simple squamous of the sinusoids. Here's the central vein, so all the sinusoids are directed towards the central vein, all right? Kind of a larger view. Here's a hepatic portal triad. So we have the large, thin-walled branch of the hepatic portal vein, a smaller diameter but more layers of smooth muscle given the radius compared to the amount of smooth muscle for the diameter of that. And here, it's a little bit like the macula densa. Here's the bile duct. Okay. So let's take a look at this model. It's made of plaster, so it's really heavy. About 15 pounds. Maybe 12, but feels like 15. Okay? So imagine that this is taken as, so on my left side, this is the central vein. Okay? So this would be one of the portal triads. And if I swung it around like that, that would be a hepatic lobule. Okay? So this is a vertical plate of cells. I'm going to put it down. So the brown cells are the liver cells known as the hepatocytes. And over here we have the branch of the hepatic artery, the red structure. The blue structure is the branch of the hepatic portal vein. And they flow together to form these sinusoids. The dark red inside is supposed to be blood. Okay? Then we have the hepatocytes, and the green lines are the bile canaliculi, carrying the bile this direction. The green structure would be the bile duct. And we actually have 4 structures here. The 4th one is the lymphatic. So that would make it a quadrad instead of a triad, so we just don't talk about it, all right? So the portal triad is the 2 blood vessels and the bile duct. As this flows in this direction, it provides opportunity for the hepatocytes to take out as well as to add. So if we have albumin in our blood, it comes out of the hepatocytes and into the sinusoids, all right, or any clotting proteins. And then it flows into the central vein, which is over on this side, okay? Now, do you remember the macrophage we had in the alveoli of the lung? We have macrophages in the sinusoids. Now, typically our macrophages are in loose, irregular connective tissue. What do you think their role would be in a sinusoid? Blood's supposed to be sterile, right? So it's not there to clean up the blood. What is one of the functions of the liver or red blood cells? It recycles them. It breaks them down, and that's what the liver macrophage is doing, okay? So as red blood cells become older, near close to or past their 4 months, their membrane becomes brittle, and they're more likely to rupture. And the macrophage is there to phagocytose it and use their lysosomal enzymes to break it down. So the name of a liver macrophage, still like to use the person's name, is a Kupffer cell. So it's very unusual for it to be within the bloodstream. It's actually shown here in the model, which is incorrect. It's right here. It's shown as an integral portion of the wall of the sinusoid. Kind of looks like a fibroblast right here. It actually is within the lumen of the sinusoid, moving up and down the length, the simple squamous epithelium. It's not part of the wall like the type 2 alveolar cell would be part of the alveolar wall. So it actually moves up and down, kind of like the alveolar macrophage moves around. So it's shown, it's not very focused, but it's kind of shown right in that area. All right, so question 3. Which of these structures would be a component of the portal triad? Guess what? You have 3 answers.
And E.
Because it's a triad, right?
Yes.
All right, does anybody want more time on question 3? So on this image, which is just the other one rotated around, the 3 structures would be this one right here, this one right here, that one right there, okay? We do have other vessels, but those would be the lymphatics, which are not considered to be part of the triad, okay? They're there, they're just not, don't work with that. All right, so if we look at an EM of the hepatocyte, anybody remember from Bio 430 what the 3 classes of organelles? So the liver makes a lot of what? Just in general, the general term. Proteins, yeah, enzymes, okay? And the albumin for osmosis. And so what are the 2 major organelles? Are these proteins kept inside the cell, or are they secreted from the hepatocyte? Secreted. So we use ribosomes that are attached to ribosomal ER. And once it pinches off of that, it has to be processed by the Golgi. So hepatocytes, for their protein synthesis role, have a lot of ribosomal ER and a lot of Golgi, all right? However, the hepatocyte has another role, which is detoxification. So it's going to take drugs like acetaminophen and break it down. It's going to take alcohol and convert it to glutaraldehyde, which is what we talk about by fixing the liver, formaldehyde, before it breaks it down to less toxic material. What's the organelle that a cell uses for detoxification? No? Smooth ER. All right, so for the protein aspect, hepatocytes have a lot of ribosomal ER and Golgi. For the detoxification, they show a lot of smooth ER, okay, because they have those two major separate roles. Notice this nucleus. Anybody remember what we call the uncoiled chromatin?
Yes.
Starts with an E, and then a U. Euchromatin. Since it's uncoiled, it means it's making proteins. All right? Now, here's the plasma membrane of our sinusoid, and there's a bit of a gap. We don't really have microvilli, but a lot of microfolds to increase the surface area. So I've already erased these. May I erase the sequence of vessels over here? You guys have that for our portal unit system? Okay.
Yes, it has the dips, the
So
glands.
here would be
The gastric
one of the
septum,
simple
gastric
squamous
okay.
cells of the sinusoid. So here would be our sinusoid on this side. And here would be our liver cell with these microrugae or folds.
In the mucosa, yeah.
And so this gap between the two, they're not tucked up right close to each other. This gap is known as the space of Dissay. The liver cells are getting discs all the time. The sinusoids also have fenestrae in them. All right, so they have large openings that allow for the proteins and the lipids to move in and out. So we'll put in some gaps here. So that allows the nutrients, amino acids, the protein-covered lipid droplets to move out.
What does
Here's a
that?
larger view.
What does that say? Space of what?
Dissay, D-I-S-S-E with a little. All right, so here's the simple squamous epithelia. And here's those microfolds. And all this fluid-filled space in here are is the space of Dissay. Given the role of the liver in storing nutrients, anybody know what all these black dots are? They're too large to be ribosomes. Anybody know what those are? So after you've eaten a carbohydrate-rich meal, the pancreas releases insulin, and that encourages the uptake of glucose, where it's stored in the liver as glycogen. So it's back in the form of a starch, a polysaccharide. And all those black dots are molecules of polysaccharides, molecules of starch, stored by the liver for 4 or 5 hours later on. When your blood glucose levels have dropped, then this is broken back down and releases glucose or fructose into the blood. And a lot of mitochondria, as you can see. All right, so here is the biocanaliculates. All right, so we have one cell here with a lot of glycogen, the basal membrane, and then the gap. So again, there's no separate epithelium. This is not like a capillary or blood vessel there. The epithelium surrounding that is the epithelium of the hepatocyte. This happens to be a liver with tumors, multiple tumors. And as you're probably well aware, the liver is a unique organ in that living donors can provide lobes of the liver or even segments of lobes, and their own liver will regenerate. Okay, so living kidneys are provided as transplants because we have two, but the other kidney doesn't grow back. In the case of the liver, if a portion of the liver is donated, the recipient's liver regenerates what is missing, and the donor rather, and the recipient's liver that was donated grows to the size needed by the recipient. For a period of time, about 5 to 8 years ago, living donor program was stopped because there were too many deaths occurring across the United States. We have a family friend who almost died. She had a rare, I don't think anybody ever figured out what she had. If you had me last semester, well, you wouldn't have had me a year ago. We did a run in Utah for her because she developed Hodgkin's lymphoma from the treatment for the liver transplant. She had to have 2. The first one was a non-living donor, and by the time she needed the second one, they had lifted the restrictions, and she had a living donor for the liver transplant. So one of the signs of before she needed the liver, it wasn't cancer, it was a copper processing issue. She ended up getting a very enlarged spleen, and that was removed before they finally decided. This all happened like her first freshman year of college. She started having health issues and had to drop out between her first and second semesters. And then she was like 27 when she had the transplant. All right, so we're gonna finish up before we take a break with the gallbladder and pancreatic components that come together within the duodenum. So may I erase the board here? We're going to look at the bile ducts coming out of the liver, and then we join that to the pancreatic ducts. Okay.
Take her quiz. I'm pretty sure this is gonna happen.
So the diagram is as if we're looking through the liver rather than lifting it up quite as much, and you can see it to some extent here. We have, down here, we have there's lots and lots of smaller ducts from the canaliculi, and then from the left lobe we have a left bile duct and a right bile duct, and that's what you're seeing up here and not shown very well up there. So I'm going to start with that as I diagram it. And because bile is often drawn green, it's actually more of a yellowish green. So left. And we'll talk about the role of the bile on Wednesday. These are coming from the lobes of the same name. And then the two join together to form a short common hepatic duct. Okay. Bile is being continuously made, whether you're eating or not. It flows down towards your small intestine. And if you don't have a need for it, it backs up and is stored in the gallbladder. Okay. So a small branch leads to the gallbladder. And this is known as the cyst for bladder. We don't look at the histology of the gallbladder, but it's lined with simple columnar epithelia. And if you saw in the sheep pluck, it's very thin walled, but there is some smooth muscle there. Okay? So cystic duct. And then the cystic duct plus the common hepatic duct form make it a little bit wider, common bile duct. So when we look at the portal vein, the portal vein, on the right side we would have the hepatic artery proper on its anterior surface. And on its left side, we would have the common bile duct. So we still see that triad pattern even outside the liver, okay? So this would be the hepatic portal vein. This would be found in the edge of the lesser omentum. Hepatic artery proper. And common bile duct, okay? So this passes behind the duodenum as a common bile duct, and then actually passes through the pancreas. So the pancreas kind of grows around it. And as I indicated, the liver is constantly making bile. If you happen to eat a fatty meal, it's going to make more, all right? Plus the gallbladder will contract to add additional bile to help it maintain the increased surface area. And it enters into the small intestine. It joins with the pancreatic duct to make, well, it's a rather large term called the hepatopancreatic ampulla, which helps you remember what forms it. The shorter name is ampulla of Vater. Darth Vader, but with a T. Bless you. So that's going to pass into the wall of the small intestine. And there's a slight raise, kind of a bump there, where it empties, called the major duodenal papilla, and you can see this in the models of the duodenum. Okay, so that's bile structures, right? Right and left hepatic ducts join to form the common hepatic duct. Common hepatic duct and cystic duct join together to form the common bile duct. And the bile flows down that to enter to the small intestine. However, there is a sphincter here, a smooth muscle called the sphincter of Oddi. That will contract or release depending on how much bile is needed. Okay? So if that is contracted, then the bile is going to go back up and go and be stored in the gallbladder. Okay? So that provides an additional amount of bile if you eat a fatty meal. In addition to what's constantly draining, you have about a half a cup or so that you can add to that by contraction of the gallbladder. Okay, almost done. We'll have to let